All right, good afternoon, everyone. I hope that you have been as inspired as I have been by the sessions today so far. Again, my name is David Berman. I'm on the MDF board, and it is once again my pleasure to welcome you to another hour of exciting updates from the developers of treatments and a cure for myotonic dystrophy. A special welcome to all of those attending the conference virtually. Again, happy to have you joining us for this session. Today, we're going to hear from 5 senior leaders of companies actively involved in drug development. You'll see them listed in the program on page 16. Today, we'll learn about the progress being made from PepGen, Juvena, Entrada, NeuBase, and ARTHEx. Due to travel difficulties from a couple of our speakers, we're going to, they can't be here in person, and they kindly put together video presentations, which will be presented to the audience. Like yesterday, we will invite the first speaker to the podium. After that, each speaker will introduce themselves. We hope to have time at the end of the session for questions. If we don't, you can meet with companies at their respective tables outside. Also tonight, there is a research poster and exhibitor reception, and you'll have an opportunity to talk to the companies directly there. The session is being recorded and will be available on the MDF Digital Academy within the next month. That again is www.myotonic.org/digital-academy. And right now, please join me in welcoming to the podium Dr. Jane Larkindale.
Hi, and thank you very much for the MDF for inviting us to speak here. I've been involved in the myotonic dystrophy community for about 16 years, and just being here as a company along with this many other companies is really exciting to me, and I hope it is to all of you as well. I am thrilled to be here, and I'm really excited to tell you about some of the work we've been doing here at PepGen. Assuming I can move the slides forward. There we go. Disclaimers, they're boring, they'll put you to sleep. Please don't shoot me with your elastic bands. They may be used as a slingshot, but please don't use them on me. Standard company disclosures. So who are we? So we're, PepGen is a small company. We're based in the Cambridge, Massachusetts area. We're moving to a new lab in Boston towards the end of the year. We're currently somewhere around about 40 people. You can see our entire company at the beach there on the slide. So we're quite small. Our focus is entirely on neuromuscular diseases at the moment, with our lead program in Duchenne muscular dystrophy and our second program in myotonic dystrophy type 1, which is what I'll be talking about today, obviously. So what is our product? We call them enhanced delivery oligonucleotides. What does that mean? Really, our product has 2 different parts. One, which is all of our programs, which is this peptide. A peptide is a small piece of a protein, and that's what allows delivery of the therapeutic part, which is the oligonucleotide. So really, our drug has 2 parts, a peptide, a little piece of a protein, an oligonucleotide, which is a kind of nucleic acid, which and the peptide delivers the oligonucleotide to where we want it in the human body. There we go. Here's our pipeline. As I mentioned, our program in Duchenne muscular dystrophy is our lead program. We're completing a phase 1 safety study in Duchenne muscular dystrophy right now, which will be hugely informative also for our follow-on myotonic dystrophy program, which we hope will be in the clinic next year. Our plans are first half of next year for our first study in myotonic dystrophy type 1 patients. What you do not see in here, and I know many of you are very passionate about, is myotonic dystrophy type 2. It is something we're interested in, but it is further back. And as I explain our technology, you will understand that why our current drug won't work in DM type 2, but the same general concept could. So I don't think I need to explain the first half of this the slide again, because I think you all know that myotonic dystrophy type 1 is caused by this repeat sequence in the DMPK gene, which binds to it. It prevents another protein binding. It binds another protein, muscle-blind type proteins, which are needed to produce proteins normally. What PGN-EDODM1 does is binds to that repeat sequence. That means the muscle blind can't bind. It should come free. It should be available in the cell to do whatever it normally does. And proteins in all systems of the body should be able to be produced relatively normally. So that's what we're trying to do. That's what our EDO is the goal of the EDO is to basically get to the root cause of disease, prevent muscle-blind and similar proteins binding to that repeat, and allowing normal cell processing to go on. I don't need to tell you this. DM1 is multisystemic. You live with a lot of different symptoms. Different people have different symptoms in different parts of the body. I think what is most exciting about our EDO technology, about this peptide, is it delivers these oligonucleotides to all kinds of tissues. We affect skeletal muscles, things like the diaphragm that affects breathing, the muscles that you use for walking and lifting your arms. We get there. We get to smooth muscle. That's things like the GI tract, your digestive system, breathing. We get there. We get to the heart. We get really quite good expression of the heart. And we even get some into the brain, which I think for myotonic dystrophy is perhaps the most exciting, that IV infusions will get this drug into the brain as well. So this is something we're really excited about. Where are we? Well, like all drug companies, we've done a lot of work in cells and mice and monkeys and all sorts of things that aren't human beings, but the data is looking good. I'm going to show you some of the data we have from patient cells. I'm going to show you some of the data we have from a mouse model of myotonic dystrophy, the HSA mouse. I'm not going to show you our monkey and mouse safety data because safety data is hopefully really boring. The only time you get unusual results is not a good thing. So I'm not going to be talking about the safety data much because it's not complete yet. So this is data from human cells from somebody with DM1 that happens to have 2,600 of CTG repeats. So it's a pretty big expansion in these cells. What you can see in the gray, on the gray side, there's a black box with some white arrows in it. Those white arrows are pointing at little things called foci, or little clumps of muscle blind and other stuff in the cell. You probably can't see the little red dots. These slides are always a problem that way. But if you look in the next column where it says PGN, EDO, DM1 in light blue, you can see there are far fewer of them. And that's what we're trying to do, is break up those foci, let the muscle blind go loose and allow it to do what it's supposed to do. And then if you go to the far side of the slide where it says missplicing correction, what it's showing is an increasing amounts of our drug, basically correcting that splicing that goes wrong in myotonic dystrophy. So with increasing doses, we're reducing those clumps of muscle blind, we're freeing the muscle blind, and it's doing what it's supposed to do in the cell. That's really exciting. Unfortunately, you can't measure symptoms in a cell, it's just a cell. We're lucky enough to work with the HSA mouse, which Charles Swonson's lab developed, which is a mouse model of DM1. It's not a perfect model. It really only has repeats in skeletal muscle. But what it does have is myotonia, just like people with DM1 may have. So again, here we went into the HSA mouse. We looked at splicing, ATP2A1 and CLCN1 just happened to be two specific transcripts. You can see, again, just like in the cells, increasing doses improves the splicing, gets it back to where it should be. But maybe more exciting is looking on the other side where we looked at myotonia in these mice. And again, as we increase the dose of PG and EDO DM1, we essentially reduce the myotonia to zero. And I'm going to show you this on the next slide. As a single dose eliminated the myotonia in this model. I'm going to ask Clede to run the first video, which is an untreated HSA mouse. What I want you to look at is the rear legs of this mouse, the hind legs. When you pinch the back of the mouse, those legs become myotonic, and you can see them dragging behind this poor little mousey with his legs dragging behind until the myotonia resolves. Now, if you look on the other side, our treated HSA mouse, you can see here, he pinches the rear legs of the mouse, and there's no myotonia. The mouse runs around perfectly normally. And this is a nice phenotype you can measure in a mouse. As I say, the mouse doesn't have all the phenotypes of myotonic dystrophy, but you can very clearly see that a single treatment with EDO-DM1 corrects this myotonia. I don't have the slide in here. We've looked at this over time. We know that the splicing changes that we see persist over about 24 weeks in this mouse model. So a single dose can last a long time. That doesn't mean we're only going to be dosing every 6 months. So 6 months in humans, we're still working out what the optimal dose in humans will be. But this is has a very persistent response to treatment. So that's really cool data, but people are not mice. We need to test this in humans. That's where we need to go. I'm pretty sure nobody out here is worrying about a mouse with myotonia. We are planning our first clinical trial in people with DM1. That will be in the first half of 2023. And we're really excited to see how it goes and hopefully get you all engaged in this development program. I've just presented in about 5 minutes, years, and really decades of research. Our research comes out of the universities of Oxford and Cambridge in the UK. Mike Gate in Cambridge developed the peptides. Matt Wood in Oxford did all the early testing in biological systems. And our partner Dennis Sperling in Paris has done a lot of the mouse experiments. And all that data has come together to show us that we can, at least in animal models and cell models, disperse the foci, free the muscle blind, clean up the splicing, and at least in a mouse model, correct myotonia. So we're really excited to get into the clinic. What are we doing now? Well, there's that left box in 2018 to 2022, since really, which is the founding of PepGen and the timing of our company. We've been working on some of this preclinical data. This year, we've had community advisory boards and clinical advisory boards. I know many of people in the audience have spoken to my colleagues and I as we talk to you about what should our clinical trials look like, what should we be measuring, how do we make it easy for you guys to come and be a part of our trial, what's too long a clinic day, how do we support your travel best. We need to work with the community. We're partners in this. We're just one spoke in a hub of activity that is needed to get a drug to market. So we've started that work. We've started talking to you, the community, and there's going to be a lot more of it in the next little while. Next year, we hope to initiate our first clinical trial, and which we'll read out in 2024. And then we'll be able to tell you much more about how this drug works in human beings, which will be really exciting. So just to come back to the community, I mentioned that we're one spoke, you guys are another spoke, the clinicians are another spoke, we all come together to make a drug. I just want to really draw attention to the fact that where we are right now is a critical point to talk to you, the community. We've been having some ad boards. I've had a lot of conversations. We want to have more. If anybody listening to this wants to tell us something that we should know about running a clinical trial in myotonic dystrophy, come find us. Our table's just out there. Come tell us. We want to make this as good an experience for you as it is for us. We want to make sure we're measuring things that are important to you. We want to hear how we can make it easier for you to participate. So please do come and talk to us. We're happy to answer questions and would really like to hear from you. And with that, much thanks to the Myotonic Dystrophy Foundation. Really happy to be here. My colleague Elena is in the back corner. We're both around. Our email addresses are there. Contact us. Let us know what's going on. Give us your thoughts. We're really excited to work with you as we move into the clinic with this drug. Thank you.
Hi, everyone. It is a pleasure to be here virtually at the Myotonic Dystrophy Foundation annual conference in order to share a bit more with you about Juvena Therapeutics and our mission to map secreted proteins to unmet medical needs, resulting in a pipeline of protein-based therapeutics for rare and chronic degenerative diseases with a strong lead program for myotonic dystrophy type 1. My name is Hannati Youssef, and I am the CEO and co-founder of Juvena Therapeutics. I'm also a scientist by training with an expertise in the biology of aging and mechanisms underlying tissue degeneration. And I'm so excited to be here today to share more about our lead program with you. And so at Juvena Therapeutics, we are decoding secreted proteins for better human health through a machine learning enhanced biologics drug discovery and development platform that is enabling us to unlock the therapeutic potential of secreted proteins and accelerate their development into life-saving therapies for multiple diseases. We have, in the last 4.5 years since our founding, not only built up a computationally driven platform for mining proteins secreted by stem cells and building a proprietary library of therapeutic candidates, but we have validated that platform with 23 hits to date across neuromuscular, skeletal, hepatic, therapeutic areas with an advancement into metabolic and inflammatory. And we have an advanced lead protein therapeutic candidate for myotonic dystrophy type 1. We have achieved these results by fundraising and securing about $49 million to date in venture capital, as well as NIH and CIRM grant funding. At Juvena Therapeutics, we are highly focused on mining and bringing to light the therapeutic potential of secreted proteins, which, while historically difficult to screen, are an underleveraged class of biologics with proven therapeutic potential. Think of drugs, for example, like insulin or human growth hormone or PepGen. Secreted proteins can communicate naturally to induce all biochemical reactions in the body, including, for example, our response to infection, proteins that can activate immune cells to launch an immune response and fight infection, for repair processes, for binding to receptors and stem cells to promote their activation, and ultimately improve differentiation, regeneration, and repair. Secreted proteins are the body's natural signaling molecules made by our cells and tissues in order to instruct them to do all sorts of phenotypic reactions and responses to promote health span. And so natural signaling molecules are highly specific to their targets. They can often bind multiple receptors to induce complex and very potent signal transduction cascades, ultimately leading to the desired phenotypic response. And they're often safer than small molecules, gene and mRNA therapies, and in comparison, especially to the gene therapies, they're cost-effective. We at Juvena Therapeutics are starting with our first generation library, which is derived by mining proteins that are secreted by human embryonic or pluripotent stem cells. Embryonic stem cells have the capability to form every tissue in the body, an entire human being. And my colleagues and I had extensively shown prior to the founding of Juvena, and even more extensively at Juvena, that we can isolate these proteins and purify them. And when added onto a variety of age and degenerating adult tissue types, they can promote improved regeneration, such as improving muscle regeneration and reducing fibrosis, or improving the ability of chondrocyte precursors to form better cartilage formation, and improving their viability when isolated from human osteoarthritic knees, or promoting the viability of cortical neurons in the face of A-beta. So it's these disease-modifying phenotypes, these regenerative phenotypes, that got us at Juvena excited about establishing a platform to identify key drivers of cell or organ-specific rejuvenation that we can then translate and engineer into protein-based therapies for a variety of degenerative diseases. And so we have a 5-step process where we start with our proprietary library of therapeutic proteins enriched in the secretomes of stem cells. We then use bioinformatics, machine learning, to rank order those proteins for their ability to induce a disease-modifying phenotype in a cell or tissue target of interest. We then screen those candidates in vitro human phenotypic screening assays that we've established at Juvena, followed by moving into extensive in vivo target validation. And once we show efficacy in a variety of different animal models, we move on to selecting the optimal candidates to move on to do custom protein engineering in order to establish both fusion proteins and antibodies that can modulate the pathways we're unraveling through our platform in order to promote tissue regeneration and repair. We have built this platform by first focusing on neuromuscular diseases, where it was our goal to identify multiple drivers of aged or dystrophic muscle regeneration to improve regenerative capacity, metabolism, ultimately strength and function, in order to treat a variety of muscle wasting diseases, ranging from the natural aged muscle degenerative disorders to dystrophies such as myotonic dystrophy. And this has led to us identifying and moving towards lead optimization for 3 candidates in particular for the diseases aforementioned, as well as several early hits across different therapeutic areas. And we are now moving, with the support of the California Institute of Regenerative Medicine and the National Institute of Health, forward with a fusion protein that we engineered that we're now developing for myotonic dystrophy type 1 and initiating investigational new drug enabling studies. In particular, our fusion protein works by targeting the regenerative signaling pathways that are downregulated due to the mechanisms of the disease. Specifically, we are enhancing and restoring AKT signaling with significant crosstalk with sphingosine 1-phosphate and BMP signaling in order to promote and restore cellular homeostasis, thereby leading to improved muscle differentiation, the maturation of fibers, improved metabolism, and ultimately survival and strength. In this example here, we took our fusion protein, and when we added it onto primary muscle precursors isolated from persons that donated muscle biopsies, persons with myotonic dystrophy type 1, we note that our protein can promote the survival as well as regenerative capacity of these muscle precursor cells, forming these multinucleated fibers, and doing so just as good as precursor cells that are isolated from healthy young individuals. We further went on to test our candidate in a variety of muscle wasting models, ranging from the natural aged model to glucocorticoid-induced atrophy, to an acute injury, and looking at a variety of different muscular dystrophies. And in all mouse models to date, our therapeutic candidate JV161, when injected subcutaneously underneath the skin, had the capacity not only to enhance muscle regeneration histologically, but to result in improvements in a variety of different performance metrics, from treadmill performance to grip strength, distance run, endurance, force measurements, and others. Here is a snippet of some of the data where you're seeing an impressive improvement in forelimb force, in distance run on a treadmill, in the cross-sectional area, and in regenerative index, meaning new fiber formation following acute injury across these chronic disease models. We have also shown impressive efficacy in a severe Duchenne's muscular dystrophy model. And most recently, we set out to establish at Juvena a DM1 mouse model that really mimics one of the greatest hallmarks of the disease, which is the muscle wasting phenotype. And so we licensed a TRED T960I mouse model from the Cooper Lab at Baylor College of Medicine and crossed that model with a pan-TET inducible mouse model, so that with the bitransgenic mice, once they're fed doxycycline, we see a reduction in grip strength compared to normal fed mouse that didn't have the DMP kinase gene with the repeats turned on. And even when you only have one copy of the mutated gene, you get a reduction in grip strength, but not as strong when the TET promoter that is inducing the gene is removed from the mouse. When it's just a transgenic mouse, you're not really seeing a difference in grip strength after doxycycline feeding. And here on the right, you're seeing a comparison of bitransgenic mice with mice that cannot get the gene turned on with doxycycline feeding to a mouse that only has one copy, and you can see that reduction in grip strength. And so we took this cohort of mice that were doxycycline fed for 13 weeks, split them into two groups in order to see if JV161 subcutaneous administration could lead to improvements in this pilot study of the histological outcomes of muscle degeneration. And in fact, really excitingly, what we see in this animal model is an increase of the presence of fast twitch fibers in JV161 treated mice compared to vehicle control, as well as an enlargement of the cross-sectional area of mature fibers. This data is very promising, and we're in the midst now of performing a large-scale study, high-powered study, in order to really measure the functional outcomes, as well as to continue with our validation of this pan-TET inducible mouse model. And we are in the midst of completing manufacturing, non-GLP safety tox studies, headed towards a conversation with the FDA with the goal of achieving our investigation in a drug in 2024 and entering into a combined phase 1/2 clinical study where we are going to perform a battery of both functional performance metrics with adult onset DM1 patients that enroll, as well as looking at biomarker profiles of improved metabolism, the patient-reported outcome, and histological metrics of muscle regeneration and repair as well. With that, we'd like to thank our advisors, and in particular, our clinical advisor, Dr. Nick Johnson, for supporting our lead program and the development of the preclinical program and clinical endpoints that we will be pursuing for JV161 treatment of DM1. And with that, I would like to thank you for your time. Happy to answer questions.
Good afternoon, everyone. Thank you for sticking around. This has been a truly amazing conference. My name is Michael Oldham. I'm one of the medical directors at Entrada Therapeutics, and two colleagues of mine, Reagan and Aji, and I are here. This is our first time at this meeting, and it truly is, inspiring and informative, and the conversations we've been able to have with all of you over these few days is just really exhilarating. Because this is our first time at this conference, I wanted to spend a couple of minutes introducing you to who Entrada is, talk a little bit about what our platform is and our clinical development program for myotonic dystrophy type 1. As Dr. Larkindale said earlier, the lawyers make us put this in, so lots of boring words, disclaimer saying that I'll be talking about things that are called forward-looking statements, so they are subject to possibly changing based on conversations we have with regulatory authorities like the FDA, for example. Entrada was formed in 2016 as a company with a commitment to bring therapies to patients who were in high need of new options for the symptoms that they had. Our team of scientists have been working over this time on a platform that we call EEV, or an endosomal vehicle escape platform. And I'm really excited and delighted that we are going to be advancing two different drugs over the next year and a half in the neuromuscular space, one of which, as I mentioned, is in DM1 that we'll be talking about more today. We truly believe that a key component of this process is, really partnering with communities, with clinicians and healthcare providers and patients, and certainly are really happy to be able to be involved with you all here and with the Myotonic Dystrophy Foundation. I do want to also just pause for a moment and commend the foundation for all of the amazing work that they've supported and promoted over the last few years. As was discussed in the opening session yesterday by the board chair, and then again today, I think, it's just incredible how much has been able to be accomplished during a time of really high stress and unprecedented kind of living throughout the world. And also want to thank everyone for that, because it is critical to driving this process of drug development and really allowing us to advance therapies more quickly out to all of you. We also are really excited to be partnering with, as I mentioned, clinicians and healthcare providers and patients, as well as the advocacy organizations. We at Entrada, along with several other companies who are here, are proud to be a part of the Myotonic Dystrophy Clinical Research Network. And I certainly want to thank the different collaborators of that network for all of their hard work, and in particular, the patients that are involved in the natural history study, or NDM1, that the network is sponsoring. Again, similar to the work that the foundation has done, participation and natural history studies like NDM1 are really vital to helping all of us as who are developing drugs to understand the disease better, understand what individuals live with day in and day out, and how to better design our studies and our trials to have meaningful endpoints and hopefully again bring new therapies to you in a quicker way. A lot of what I'm about to talk about are things that you may have heard already over the course of yesterday and today in terms of some of the fundamentals of the problems and challenges that we face when we develop drugs and also the way that we're developing them. To me, this is actually really exciting because of all of the work that has already happened in terms of developing models of myotonic dystrophy type 1, for example. It really gives all of the different companies opportunities to test out new potential drugs, again, to bring those forward into the clinic. So while some, I think, may be a little frustrated to kind of hear the same thing over and over, I actually find it really, really exhilarating and just speaks to all of the hard work that has been done over the years leading up to now. One of the things that we know about, medicine and diseases in general is that there's a lot of drugs out there, but unless you get them to the targets or where they need to be, then they can't do their jobs. And even if you get it to where it needs to be, the drug might be broken down, metabolized, or cleared away too quickly to actually exert its function. And so one of the solutions is the platform that I had mentioned previously that Entrada has developed that we call EEV, or Endosomal Escape Vehicle. And early studies have shown that by linking this EEV, which is depicted by the small round yellow gold in the figure, linking that to a certain type of drug, in this instance, an oligonucleotide, the complex or the conjugate of those is able to get into the cells and where it needs to be and actually stay there longer to carry out the functions that it needs to. One of the reasons that you've heard quite a bit about oligonucleotides and why this is a type of specific therapy that we also are excited about really has to do with some of the fundamentals of how diseases arise. We know that, for example, genes are located on the DNA and are transcribed to RNA. And then that RNA is translated into proteins that all of our bodies need to function. And at any point along this pathway, there can be a disruption, a change, or a problem that arises that leads to certain symptoms. And that's where a therapy like an oligonucleotide can come into play, because it can act higher up in this whole process to have a positive effect downstream. Again, as you all know very well and have heard about extensively over the last 2 days and previous to this, one of the main issues in myotonic dystrophy type 1 is the DMPK gene and the excessive number of repeats, and how this can lead to the formation, as you see in the middle part of the figure, the elongated messenger RNA, something that is often described as hairpin loops or elongated mRNA. And one of the things that can happen is that the certain types of proteins, in this case, something called MBNL, can get stuck there or sequestered, which we've heard a good deal about again at this conference. And when that happens, what's formed is called a nuclear foci. So that sticking of the MBNL to that messenger RNA. And when it does that, the MBNL is not able to carry out its normal functions. It's not circulating through the body and interacting with other proteins like it's supposed to. And then this results in many of the symptoms that individuals experience. So weakness, myotonia, possibly even the heart manifestations. And so we at Entrada hypothesized that if we were able to get our drug, which we call ENTR-701, where it needs to go, that it will plop down on that messenger RNA and allow those MBNL protein to continue to float around and carry out the job that they're supposed to do, which is interact with other proteins, like I mentioned. So we tested this hypothesis again in many of the same types of models that you've heard about yesterday and today, both cells and mice models. You can see in the middle here, one of the mouse models that we tested. The top panel there is essentially an untreated mouse, and all of the red dots represent those nuclear foci that I mentioned. So the MBNL protein that's clumped and stuck to the messenger RNA. In the mice that were treated with a single injection of ENTR-701, is a muscle biopsy from those mice is depicted in the lower part of the panel. And you see a significantly less number of those red dots, which represents much fewer of those nuclear foci. And in this particular image, this is just showing one specific muscle, but we looked at this in multiple different muscles and throughout different parts of different body systems. We also were really excited to find not only a reduction of these foci, but that other changes were seen downstream that demonstrated those proteins that I mentioned were not getting clumped up and were able to continue and carry on to do the job that they were supposed to. While this is obviously really important to see changes in cells, we also wanted to look at whether or not there were any meaningful changes in the mice. And this is very similar to what you saw in a previous presentation of a similar type of mouse model, where on the left, when the little guy is pushed on his hind legs by the scientist, the hind legs develop myotonia, and that was in one of the untreated mice. On the right panel, you see, and I should mention thank you to the guys in the back for running these videos. When the mouse has their hind legs pressed, they do not develop the myotonia. They continue to kick around and move without developing that symptom. So armed with the results from the different cell studies, the mice studies, et cetera, we at Entrada have continued and proceeded to run several of the other preclinical studies that are needed to engage with regulatory authorities. So other mouse and monkey studies looking at safety. Sometimes these are called toxicology studies. And all of this combined, all of these data, we intend to put together in a package and submit to the regulatory authorities like the FDA within the next year, which is that next step along the path to move into setting up clinical trials of ENTR-701 in patients with DM1. So again, we are really excited to be at the beginning stages of working with each and every one of you. As I mentioned, one of my colleagues, Reagan, who's our head of advocacy, and Aji from our scientific communications, we three are here and would love to speak with you. Reagan's email is listed here. We also have a couple of posters, and the QR code on the screen will send you to a link that has a lot of our publications and other posters, including those here. As I said, we are at the start of this journey with you. And so if there is anyone interested in telling us their story and working with us as we go down this path, please do not hesitate to reach out to us. We'd love to continue the engagement. Thank you so much.
Good afternoon. My name is Dietrich Steffen, and I'm the chairman and CEO of NeuBase Therapeutics. It's a pleasure to join you for the 2022 Annual Myotonic Dystrophy Foundation meeting. NeuBase Therapeutics has developed a platform that allows us to directly engage with genes that are misbehaving and neutralize them before they manifest in disease. We can do this through patient-friendly routes of administration, and we've shown this now against several genetic targets. And next calendar year, we plan on dosing our first patients in our clinical trial in myotonic dystrophy type 1. Now, we're all familiar with the prevalence and severity of myotonic dystrophy type 1, but I want to remind you of the pathogenic mechanism as context for how our drugs work. As we all know, there's an expansion in the 3' untranslated region of one copy of the DMPK gene that, when transcribed, forms an aberrant hairpin structure in the nucleus in the RNA. Now, that aberrant hairpin sequesters critical splice proteins, as illustrated in the middle panel of this slide. And when those are sequestered or trapped in this toxic structure, that results in an inability for many other mRNAs to be processed correctly. Now, what's important is that in preclinical animal models with this disease, aspects of the disease have been shown to be reversible when this toxic structure is disrupted. So we're all familiar with the resolution of myotonia in several of the transgenic animal models, but there is also evidence from Baylor, for example, that the cardiac aspects of the disease may be reversible. And most recently, an elegant paper from the Paris group illustrating that aspects of the cognitive deficits may be reversible. And we're quite hopeful that this may be translatable to human patients. Now, how does our molecule work? First of all, we've designed this to be a whole body solution. So I'll show you data that a patient-friendly route of administration, such as an intravenous injection, can get into not only skeletal muscle, but also cardiac muscle and the brain. And that gives us hope that we can address all of the major aspects of the disorder. Secondly, we believe that subsequent generations of our compounds can be delivered via subcutaneous routes, even more patient-friendly. Now, importantly and differently, we only engage with the mutant copy of the DMPK transcript and resolve those toxic nuclear aggregates, but leave the wild type copy of the transcript around under the rubric that transcript is important and its protein product is important for healthy function. And so that's different. So brain exposures and maintenance of the wild type transcript are important differentiators of our strategy, which is predicated on invading that toxic mRNA in the nucleus and sterically displacing muscle-blind proteins, or all of the splice proteins, such that they can go resume normal healthy processing of downstream mRNAs. Now, mechanistically, we've proven that this strategy works repeatedly. I will show you two slides to illustrate this point. This is data illustrating that a single intravenous injection into the gold standard transgenic animal model, which is the HSA-LR model developed by the Rochester group, not only engages the target mRNA and statistically significantly reduces this toxic mRNA level beginning at day 21, but also almost completely rescues healthy processing of, in this case, 3 downstream transcripts. And so I'll just briefly dwell on this. As you can see in blue is the level of incorrect splicing in the transgenic animal untreated relative to healthy animals in black. Now, when we dose with our development candidate, which is named NT0231, we can say at about 21 days, we begin to statistically significantly rescue cassette exon use towards correct use in black. And that continues to mature over the course of 6 weeks until by day 42, we have complete rescue, in this case of ATP2A1, relative to healthy background animal strains. And that picture is similar for other transcripts that are characteristically misspliced in the disease. And so from a target engagement and splice rescue perspective, the compounds work beautifully in this transgenic animal model. And that includes correcting splicing of a transcript named CLCN1. This is the skeletal muscle chloride channel. It's thought that incorrect splicing of this channel is directly causing myotonia. And what you can see when we rescue splicing here in this CLCN1 transcript, that allows the resultant protein to be produced normally, insert into the myofiber membrane. And by day 28, we begin to see functional correction of myotonia. And by day 35, we have statistically significant correction of myotonia in this transgenic animal. I should note that this is measured by direct relaxation assays. And so this is an assay named the plantar flexor torque assay. So we believe that this is translatable forward as an endpoint in humans and different than measuring EMG corrections. Now, how does this work with multiple dosing, and can we lower the dose? So these are experiments that are ongoing. But what you see here is that if we, rather than a single IV dose, begin to administer weekly, we can begin to reduce the dose 20-fold in this high bar transgenic and still maintain statistically significant correction of splicing of CLCN1. And then that also translates into statistically significant rescue of myotonia. And so we're exploring these different regimens, but importantly, we do have headroom to reduce the pharmacologically active dose in this animal model. Now, this sets the stage for our IND filing and dosing our first patients in calendar year 2023. We do believe that we have a therapeutic index to be able to move the program forward and again, provide a differentiated solution to patients. And so what you'll see in the remainder of this calendar year and into early next calendar year is data illustrating the pharmacologically active dose in human patient-derived myotubes, which will set the pharmacologic floor for the clinical trial, the maturation of the dosing regimen and cadence of dosing, all of the GLP toxicology work that's necessary in order to file the IND, and moving our manufacturing into GMP to fuel the trial. So that's the work that's ongoing now. Positioning for the phase 1/2 trial in affected individuals is participation in the large national natural history project that many of the sponsors are involved in, as well as establishing clinical proof of concept in that phase 1/2 with a biomarker-based approach. So I hope that gives you an update of where we are with respect to bringing what we believe is a differentiated and patient-friendly option to the clinic and ultimately to market. Remember, our hope is to be able to provide a solution that not only addresses the skeletal muscle issues that patients suffer from, but also the cardiac and respiratory issues. And we've illustrated nice exposures of our compound after intravenous injection in all three of those muscle groups, but also addressing the cognitive deficits that both adult and pediatric patients complain of, which are significant. And we believe we're the only one of the first class of companies going to clinic that have the ability to potentially address those cognitive deficits, and fingers crossed those are reversible. We're also pleasantly inclined with respect to the preservation of the wild type DMPK mRNA and the resultant protein, because we believe that that's important, and that allele selectivity in addressing the disease is I think, going to be an important aspect to our option moving forward. So please reach out to us if we can provide any further information. You can also see the resources that are available on this slide, either at our website, NeuBaseTherapeutics.com. Please follow us on Twitter or LinkedIn. And if you'd like to speak with us directly, we're always available at info@neubase.com. We'd love to keep you up to date on our progress as we move closer to our planned clinical trials next year. So thank you all for the opportunity to give you an update, and I wish you a wonderful rest of the conference.
Here it is. Hello, everyone. I'd like to thank you for granting me the privilege of speaking with you. And thank you for staying for this last of the industry updates. I know it's been a long haul. Hang on. How do I advance the slides? Oh, this is the slide advancer. Sorry. Just grappling with the technology here. I'd also like to thank the MDF for allowing me to speak to you today. And I would like to thank my colleagues from the other companies because they've laid a lot of the groundwork. So I don't need to go over a lot of things. So I'm going to focus in my talk on sort of what differentiates our company and our approach to myotonic dystrophy. I've got a very brief disclaimer, but essentially says the same as everybody else's. So who are we at ARTHEx Biotech? So we are a very small company. We're based out of Valencia, Spain. Although I'm not, but most of the people in the company are based in Spain, but we have a very global outlook. We started as a spinoff of the University of Valencia, where in the lab of Ruben Artero, they were looking at ways of dealing with diseases with unmet medical needs, and in particular neuromuscular diseases, and specifically myotonic dystrophy as the first focus. And the idea was to develop treatments that blocked or limited the activity of microRNA. Now, you've heard in this conference about DNA, RNA, messenger RNA. So what is microRNA? Well, microRNA are small oligonucleotide molecules that serve as regulators of function of other molecules, other RNAs. They don't actually code a protein like the messenger RNA does, but they still play a very important role, and we're just starting to discover that role and how they may play a role in various different pathways that lead to disease. So our focus is on anti-microRNA treatments. And we have, like every other company, we have a platform. Our platform is called the Enhanced Anti-Mir Delivery. So a mir is a microRNA, an anti-mir is a molecule that blocks the microRNA. We call it the entry platform. And basically what we do is we combine an oligonucleotide that is complementary to the sequence of the microRNA we want to block. And we combine that with a hydrophobic conjugate. This is a molecule that ensures that we get delivery to the tissues we want to get delivery to. And that entity, that little hydrophobic conjugate will change based on the disease and which tissues we want to focus on. So when we did this for DM1, we came up with a product called ATX01. And like the other companies, I think everybody's working on the same thing. We're working on getting a molecule that we think will play a role in reversing or preventing the disease, but also making sure that we deliver that molecule to the right tissues. And we've been able to show that we have achieved both of these goals. So I'm not going to tell you what DM1 is because you know way more about this than I do. But I'm going to show you a little bit about how our drug works in the context of the pathophysiology of DM1. So you all know about how the DM1 is caused by an expanded repeat of greater than 50 in a non-coding region of the DMPK gene. And we know that at some point this leads to reduction in MBNL protein, which then leads to abnormal splicing, which then leads to the symptoms of the disease. What happens in between, we know that there is an increase in these hairpin loops with sequestration of the MBNL. And so that is one reason why the MBNL is reduced, is because it's sequestered, it's trapped in those hairpin loops. But there's another reason that's less talked about, but has been very well established. And that's because the hairpin loops trigger an increase in microRNA 23B. So miR23B is increased by these toxic RNA. The miR23B in turn suppresses the production of MBNL protein. So your MBNL protein function is lost because of sequestration, but also because of lack of production. So it's a double whammy. So what we work on is we work on both aspects of these, and I'll show you how we do that in the next slide. So what happens is that when you, do I have a penalty here? Yes. No, it's not going to work there. So when you, pointer's not going to work, forget that. So when you block miR23B with a complementary oligonucleotide, you do two things. So first is that you de-repress that repressed production of MBNL. So now MBNL production can continue as it should. MBNL levels then increase. But also, the miR23 blockage reduces the number of toxic DMPK RNA. So you end up with an increase in MBNL protein from these two mechanisms. So that's why we call it a dual target. Reduced DMPK, and the graph on the right, and I apologize, it's very small, shows you the reduction of the toxic DMPK foci. And the bottom graph shows you the increase in MBNL protein levels. So that's the theory. Does it work in animal? Well, we've done a whole host of studies. We've done the previous page. I showed you the human cell line studies, and now we've done a host of animal studies. This is just one of them. I should also mention we have a poster where we're talking about some of our human cell line studies that I'd be delighted if you came by and spoke with me about. But in the animal studies, I'm showing you one example, and this was in the HSALR model, which you've heard about. And it was a multi-dose study, and we gave these mice injections at day 0, day 30, day 60, and then we sacrificed them at day 95. And we showed a couple of things. So first of all, the bottom left graph shows strength. And if you look at the dotted line, that's normal strength in the normal mice. The gray line at the very bottom shows the reduced strength in the untreated mice. And the two lines in between show the treated mice at different doses. And so we tested a low dose and a high dose, 3 milligrams per kilogram and 24 milligrams per kilogram. Both of these achieve almost complete reversal of the loss of strength. The difference between the two doses was with the 24 milligram per kilogram, there was a longer duration of effect. So then when we looked at the muscles and we looked at target engagements, we looked at were we increasing the MBNL protein as we thought we were, and indeed we were in a dose responsive manner. So the higher dose got you more MBNL. And we also improved, we also had splicing rescue with low dose and even greater with the high dose. I should also say there was also improvement in myotonia as well. So subsequent to this, oops, why is it not working? Okay, great. So subsequent to this, we've done a number of different studies. We've almost completed our toxicology package, which means the studies that we do in animals to make sure that it's safe and that we can proceed to administer the drug to humans. And that is all looking extremely good. We had a meeting with the with the FDA, pre-IND meeting with the FDA last September, and we know what we need to do to start a study in humans. We have just submitted an application, a dossier to the European Medicines Agency with some questions we want to ask and get some advice on the program. And we're gearing up to get started with our clinical trial early next year. The study is going to be done in the US and in Europe and hopefully Canada, which is my home country. So we're hoping to make this a very global clinical trial. I'm not going to talk specifically about the clinical trial, but happy to answer any questions. And we have already spoken to many of you to get input on the design, and we will continue to do so because obviously it's very important to get your input on this. But the study is going to be very similar to what you've seen from other companies with a single ascending dose followed by a multiple ascending dose, followed by an open label extension. And we are going to look at the muscular symptoms. We're going to be looking at things like myotonia, strength, muscle functions, like many of these same outcome measures that others have looked at. We are also, because, and I didn't show you this, but we also have shown target engagement in cardiac muscle and in brain. So we are also going to be looking at some of the CNS symptoms such as cognition, apathy, that type of thing. We're going to be looking at even the eye, hopefully, and a number of other symptoms, because we do believe this drug has a potential to affect not just muscle. So I said I was going to focus on what made us a little bit different from some of the other treatments. And isn't it wonderful that we have all these different treatments? You know, I think this is just a great time to be in doing research in this space, because there are so many different options. And the reality is that in drug development, you need to take what we call many shots on goal. So we need to look at many different mechanisms. Again, our treatment is a little bit different from the others, and each treatment has its own differences, because number one, we're an anti-microRNA, and I think we're the only anti-microRNA treatment for myotonic dystrophy. We have this dual mechanism of action. One thing I didn't talk about is that the miR23B sequence is very well conserved, meaning that if you look at the sequence that we're targeting, if you look at it in mice, rat, dog, human, it's the same sequence. And this means that the anti-MIR that we're using as treatment is going to be the same one that we used in mice, we will use in humans. And this is important because a lot of times in drug development, you have to change the molecule, you have to tweak it slightly, because the sequence you're targeting is not conserved. I mentioned already that we have effect in heart and central nervous system, not just muscle. I didn't mention because we're not targeting DMPK directly, we're targeting MBNL, we could potentially have an effect in DM2 as well. So we're started some studies in cells of patients with DM2, and we're very hopeful that we can do a study in DM2. And then we also have a very simple chemistry. And why is that important? That's important because when you have a simple chemistry, it's simple to manufacture. Simple to manufacture means low cost of goods. Low cost of goods means lower price for the patient. And we know that sometimes you can have great medicines, but if patients can't access them, it's not terribly helpful. So again, you know, we are so thrilled to be here and to be speaking with you, and we will continue to speak with you. And would love, as everybody has said, your suggestions to make this a more successful program, because we really want to improve the lives of patients with myotonic dystrophy. Thank you.